In the early 1900s, a British geneticist named Reginald Punnett developed a grid which enabled scientists to quickly determine the possible gene combinations for a cross or mating. In the Punnett square, symbols for the genes from the male parent appear along the top of the grid, while the genes supplied by the female appear along the left side of the grid. By combining each pair, all the possible combinations become known. Let's try an example. Short hair is dominant over long hair in the domestic cat. The dominant gene, short hair, is represented by a capital S. The recessive gene, long hair, is represented by a small s. What happens when a pure short-haired cat and a pure long-haired cat are mated? What is the phenotype, or actual appearance, of the cats we are studying? The phenotype, the actual appearance of the cats, is 100% short-haired. In each box of the Punnett square, a dominant short-haired gene combines with a recessive long-haired gene. All the cats will appear short-haired because the dominant gene masks the recessive gene. What is the genotype, or actual genetic makeup, of the cats we are studying? The actual genetic makeup or genotype of the cats being studied are 100% hybrid or mixed. Each cat will have a dominant gene for short hair and a recessive gene for long hair. What happens if two of the hybrid cats from the first generation are mated? What are the phenotypes, or actual appearances, of the offspring? 3 will have short hair and 1 will have long hair. What are the genotypes, or actual genetic makeup, of the cats? The genotype or actual genetic makeup are 1 pure for short hair, 2 hybrid or mixed, and 1 pure for the recessive trait long hair. It was fortunate that Gregor Mendel studied traits that were specific and did not vary significantly from one generation to the next. Mendel's pea plants were either tall or short, yellow or green, smooth or wrinkled, and so on. There was never a plant that was in between. However, scientists soon discovered that not all genes behave as Mendel predicted. The Japanese 4 o'clock is an example of this. The 4 o'clock is found as a red or a white flower. A cross between a red and white 4 o'clock should result in either all red or all white, depending on which gene is dominant. However, the cross results in all pink flowers. Neither gene appears to be dominant. Indeed, both genes appear to be expressed in the offspring, resulting in traits not found in either of the parents. Scientists call this observation codominance, or incomplete dominance. Since neither gene is dominant, scientists designed another method for designating the genes involved in the cross. Capital letters are used for the trait, for example, capital C for color, and a superscript is used for the specific trait being studied. For example, R for red and W for white. Thus, RW would be the genotype for a 4 o'clock that is pink. The Punnett square would look like this. Now you try one. The Andalusian fowl, a type of chicken, has 1 gene for black feathers and 1 for white feathers. Breeding the two results in chickens that appear to be blue. How would you represent a cross between a black fowl and a white fowl? Capital F is used to indicate feather color. The feather color of the genes contributed by the male is black and is indicated along the top horizontal line of the Punnett square by the superscript B. The feather color genes contributed by the female are both white, and this is indicated by the superscript W. The genes contributed by the female are displayed along the vertical line of the Punnett square. Finish filling in the square. Turn off the player until you have finished. This is how your completed Punnett square should look. When all possible combinations are displayed in the cells of the Punnett square, it becomes clear that all offspring will appear blue, because they each receive a gene for black feather color and a gene for white feather color. To review then, the Punnett square is a graphic way to predict what the outcome of matings will be. In the Punnett square, symbols for the genes from the male parent appear along the top of the grid, while the genes supplied by the female parent appear along the left side of the grid. By combining each pair, all the possible combinations become known. Codominance, or incomplete dominance, describes a situation when neither gene in a cross is dominant. Both genes are expressed in the offspring, resulting in traits not found in either of the parents.